Hello friends, our today's topic is the meiosis. In the last lecture we have seen the mitotic division. Now the next topic and the last topic of this chapter, that is the meiosis division. So meiosis division, it is a complex type of the cell division as compared to the mitotic division, because here is a reduction in number of chromosomes. In mitosis, if the parent cell have 46 chromosomes, then the two daughter cells also have a 46 chromosome in each cell. But in case of meiosis, if the parent cell have 46 chromosome, then the daughter cell, which meiosis produces 4 cells and mitosis produces only 2 cells. So number of cells are 4 in meiosis, and the chromosome number will also get reduced to half. Means if the parent cell have 46 chromosome, then the daughter cells in the meiosis will have 23 chromosome, means half number of the chromosome, that is haploid. So that is why it is a very complex type of cell division. It occurs only in a special diploid cell. Meiosis does not occur into the haploid cell, it occurs into the diploid cells, means the special diploid cell. Now which are the special diploid cells? In the plants, in case of plants, the special diploid cells means the spore formation. Okay, during the spore formation there is a meiosis. Then during the gamete formation in the angiosperms, that is higher plants, also there is a meiosis into the gamete formation. Okay, and in case of animals, germinal cells, in germinal cell there is a meiosis. Germinal cells means which produces the gametes. In case of humans, meiosis occurs in the testes in the male and ovaries into the female. In the testes there is a germinal cells which are present into the testes, into the seminiferous tubules. The germinal cells undergo meiosis division and produces the gamete, that is the sperms. And in the female there is a ovary, and in ovary there is a development of the follicles, different follicles, number of follicles, and in that follicle there is a developing ovum. That ovum is produced by the meiotic division. That process is called as oogenesis, and the formation of sperm is called as a spermatogenesis. All the two processes, that is the spermatogenesis and oogenesis, we will see into the reproduction chapter. So next, meiosis division is also called the reduction division. Why it is called as reduction division? Due to the reduction in number of chromosomes, means the chromosome number becomes the half. If the parent cell consists 46 chromosomes, in the daughter cell it will become 23. If the parent cell contains 40 chromosomes, then in the daughter cell it will become 20. So reduction of number of chromosomes by the half. That is why this is called as a reduction division. And mitosis is called as equational division, because there is equal distribution of the chromosome. Means the parent cell, if the parent cell have 46 chromosome, then the two daughter cells also have 46. That is why it is called as a equational division. Now another one important point with respect to the meiosis is genetic material, that is the DNA, duplicate only once, but cells divides twice. Means duplication of DNA takes place only one time, but the cell divides twice or 2 times. But in the case of mitosis, duplication of DNA is one time and the division of the cell is also one time. That is why there is no reduction in number of chromosomes. Now this meiosis completes in the 2 stages, that is meiosis first and meiosis second. Meiosis first is called as reduction division and meiosis second is called as equational division. It means that in meiosis first there is a reduction in number of chromosomes, but in meiosis second there is no reduction in number of chromosomes. So meiosis second is similar to the mitosis. It is exactly same as the mitosis. So the difference is in meiosis first. So meiosis first also have interphase, and in interphase there is a duplication of the genetic material, that is the DNA, and all the process synthesis of the enzymes and molecules which are required for the cell division which we have seen into the interphase. So that's why here I am not explaining the interphase, because we have seen already interphase. Now we are actually starting the meiotic division, and in meiotic division there are two substages, that is meiosis first and meiosis second. So first one is meiosis 1, and these meiosis 1 also have 4 substages that are prophase, metaphase, anaphase and telophase, which are present into the mitosis. So same steps are present into the meiosis 1, but the numbering is prophase 1, metaphase 1, anaphase 1 and telophase 1, because these are the stages into the meiosis 1. That is why prophase 1, metaphase 1, anaphase 1 and telophase 1. So these are the 4 stages we have to see into the meiosis first. But the difference is that the prophase 1 again have 5 substages, prophase 1 again have 5 substages. These 5 substages are very important, and this is the difference in the meiosis and mitosis. Means these 5 substages of the prophase 1 which are not present into the mitosis 1, and these 5 substages are actually responsible for the variation, because meiosis produces the variation due to the exchange of genetic material, and that exchange of genetic material takes place within these 5 substages, and reduction of the chromosome also takes place in these 5 substages. So that is why we have to study all these 5 substages, which includes the 5 substages are prophase 1 have 5 substages, that is first one is liptotiny, second one is zygotiny, third one is pachytiny, fourth one is diplotiny, and fifth one and last, that is the karyokinesis. Simply I am repeating, first one is liptotiny, second one is zygotiny, third one is pachytiny, fourth one is diplotiny, and fifth, that is the last, is karyokinesis. You can remember these stages by a short trick, and sequence is necessary. So you have to remember this short trick, that is le ZP Didi, le ZP Didi. ZP, you know that ZP means the Jilla Parishad school. So le ZP Didi, ZP is ZP Didi, ZP is the Didi. So le ZP Didi, it means that you are giving something to the ZP Didi. So that is why we are saying that le ZP Didi. It may be whatever. So you have to remember that is le ZP Didi. So le for liptotiny, Z for zygotiny, P for pachytiny, first Di means diplotiny, and second last, that is the karyokinesis. Simply you can remember from this trick, that is le ZP Didi. So now in the meiosis 1 there is prophase first. So we will see the prophase first. Prophase first is the most complicated and the longest phase, because there is all these 5 substages. That is why it is very complicated and it requires the time. This prophase first consists the 5 substages. We have seen these 5 substages, means the only names, not in detail. So it consists of 5 substages, that is le ZP Didi, liptotiny, zygotiny, pachytiny, le ZP Didi, here will be D not P. So liptotiny, zygotiny, pachytiny, diplotiny and karyokinesis. So now we are starting with the first stage of the prophase first, that is liptotiny. So now we are starting liptotiny. Listen carefully. Liptos means thin and nema means thread. Means in this stage we will observe or we will found the chromosome as a thin thread like structure. That is why this stage is called as liptotiny or liptonema. The meaning of liptos means thin and the meaning of nema is the thread. Now we will see the diagram, then I will explain the theoretical part of this liptotiny. Now in the prophase first and liptotiny, the cell have the normal chromosomes, there is a duplication of the genetic material in the interphase. So here we are considering diploid cell because this occurs into the diploid cell, not into the haploid cell. So we are starting with the diploid cell, but we are considering the suppose human cell, and in human cell the diploid condition have 46 chromosome, but we cannot draw here 46 chromosome because it will become very complicated. So we are making only 2 chromosome, or we are showing only the 2 chromosome, means 2n condition, 2n is equal to 2c, means diploid condition is represented by 2 chromosome. Here we are representing diploid condition in the form of 2 chromosome. In the mitosis we have taken 2n is equal to 4c, means diploid condition have 4 chromosomes. Here we are taking diploid condition is equal to 2 chromosomes. So that you have to remember this point. We are taking 2 chromosomes only as a diploid condition. So what will be happen in the haploid condition? It means that when 4 cells are formed and after the completion of meiosis, it will, these cells, all the 4 cells have only one chromosome, because the diploid condition have 2 chromosomes. That's why haploid condition will have 1 chromosome in each cell. So that's why you have to remember this point. We are considering 2n is equal to 2c. So now look at carefully at the diagram. This is the nucleus, this is the centriole, this is the cell membrane or plasma membrane of the cell. Inside the nucleus there is a nucleolus and the chromatin network, that is the DNA, is present. So these are the chromosomes. So there are 2 chromosomes, 2n is equal to 2c. So this is a 1 chromosome and this blue one is the another chromosome. So this cell basically have 2 chromosomes. Now what happening in this stage liptotiny is the chromatin network condensation will be start and appears as a long and a thin thread like chromosome. So that is why these are thin and long thread like chromosome. That is why this stage is called as a liptonema or liptotiny. Now next point, each chromosome consists of 2 chromatids. Each chromosome consists of 2 chromatids but appears as a single thread. It means that, look at this diagram. This is a chromosome, that is the centromere where these chromatids are attached. Now this one is one single chromatid from this up to this point. So this is a complete one chromatid and this is another second chromatid. So what is second point? Each chromosome consists of 2 chromatids, means 1 and this is second, not 4. 1, 2, 3, 4. You have to count, this is complete 1 chromatid and this is 1 complete chromatid. So that's why 1 and 2. But it appears in liptotiny as a single thread. That's why in this diagram I am showing showing only one single thread in blue also and black also. So these are the changes which are occurring into the liptotiny stage. So here we completed the liptotiny stage. Next stage of prophase first is the zygotiny. We have seen the first stage, that is the liptotiny. Now the second, that is zygotiny, or it is also called as zygonema. So zygos means the pair and nema means thread. So in this stage there is a pairing of the chromosomal threads. That is why this stage is called as the zygotiny. So this is the pairing of the chromosomes. Now which chromosome pairs? There is a pairing of homologous chromosomes. So homologous chromosome begins to pair. Now what are the homologous chromosomes? Homologous chromosomes are the pair of chromosome, in which one is paternal chromosome and another one is maternal chromosome. Means in the pair, paternal means from the father and maternal means from the mother. So in a homologous pair of chromosome, it consists one chromosome from the father and another chromosome from the mother. Suppose these are the two chromosomes, one from father and one from mother, so these are the homologous. Now not only this condition, but when such a chromosomes have the same genes at the same locus, this is called as homologous chromosome. Now what is this actually? Suppose this is chromosome from father and this is chromosome from mother. So suppose at this chromosome on this upper side there is a gene which codes for the height, that is for capital T or small t, maybe tall or maybe dwarf, but it codes for a height. And from the mother, this location also have a gene for the gene which codes for the height. So the position, that is the location, which is called as locus. Locus means the position of gene on the chromosome, it is called as locus of that gene. So locus is same for the height. Now suppose consider another gene, B gene. If this chromosome which is from father, so on the paternal chromosome the B gene is present at this location, and also in the mother's chromosome the B gene is present at this location. Then suppose on the lower side the C gene and here is also the C gene, means the same type of the genes are present on the same loci. Such a chromosome are called as homologous chromosome. So now these two are homologous because the location of the genes are the same in both the chromosomes, and one is from the father and another is from the mother. So such a chromosome is called as a homologous chromosome. So pairing is started or begins in between the homologous chromosome, means which have the same type of the genes, and this pairing is done by the complex, that is synaptonymal complex. This pairing is done by the synaptonymal complex which forms the pairing, and such a pairing of the chromosomes of homologous chromosome is called as the synapsis. So this process is called as a synapsis. So what is synapsis? Synapsis means pairing of homologous chromosome is called as a synapsis. So this is the important point which is happening into the zygotenous stage, that is the synapsis. Now, so due to the pairing, we will see the chromosome in the form of bivalent or tetrad form. So this is actually a pair of homologous chromosome. This is a pair of homologous chromosome. There are two different chromosomes, means two chromosomes. 2n is equal to 2c. We are considering the 2c. So that is why this blue and black are the pair of homologous chromosome. म्हणजे काय झाले तर पेअरिंग या ठिकाणी झालेले आहे. ठीक आहे. Now in the next look at this diagram. This is called as a bivalent. So this is a bivalent or also called as tetrad. Now why it is called as tetrad? Because each chromosome in the first stage, that is zygoteny, we have seen each chromosome have two chromatids, but it appears as a single thread. So here I am showing the complete two threads. One complete thread, chromatid, and this is second chromatid of one chromosome. So two chromatids. Then here is also one and two chromatids. So this blue also have two chromatids and black also have two chromatids. So total will become 4. So 1, 2, 3 and 4 chromatids. That is why it is called as a tetrad. That's why it is called as a tetrad. So it will look like this. Because each chromatid, suppose this one chromatid have produced the second chromatid, because due to the duplication of DNA, same copy of DNA is produced and which forms the chromatid, another chromatid on the same chromosome. So this is the actual, and here the centrioles which are start to migrate towards the opposite pole. So this is a zygotenous stage. In zygotenous stage, the important point is the synapsis. Now what is the synapsis? The pairing of homologous chromosomes. Simply, that is the synapses. Now the next point or the next step is the Pachyteny stage, third stage, Le Z P. Now P for Pachyteny or also called as Pachynema. So this Pachus means thread and nema means Pachus means thick and then nema means thread. So there is the formation of thick threads. That is why this stage is called as a Pachynema or Pachyteny. Now we will see the enzyme later. Look at the diagram. There is a pairing of homologous chromosome, and now this homologous chromosome we will see in the form of tetrads. So one chromatid, second chromatid, this is the third chromatid, this is the fourth chromatid, this is nucleolus, and now centriole is migrated to the opposite poles. So now in the Pachyteny stage, the important topic is the crossing over. So now what happened in Pachyteny? There is a crossing over in the homologous chromosome. Pairing of homologous chromosome takes place into the zygodyny stage. Crossing over in homologous chromosome will take place into the Pachyteny stage. So what happened actually, the crossing over means, or what is the crossing over? So simply the crossing over means the exchange of genetic material between non-sister chromatids of homologous chromosome. Now you have to focus on the non-sister chromatids. What are actually the non-sister chromatids? Now look at this diagram. These and these are the sister chromatids. These and these are sister chromatids. Why these are sister chromatids? Because during the duplication of the DNA, the chromosome have only one single thread. As the duplication takes place, suppose here is a DNA and these also have DNA, because chromosome is made up of DNA. So what happens? This DNA gets duplicated. Here is also duplicated. So here is a formation of the new thread. Now this black one is produced from this blue. So that is why two are the sisters. And same in the case of this side, this blue has produced this black DNA. So that's why this thread or chromatid is the sister. So these two are sister, these two are sister. But this and this not are sister, this and this are not sister. Means in the case of these chromosomes, these are sister chromatid, these are sister chromatids. These two black are sister chromatid, these two black are sister chromatid. But this black and this blue are non-sister. These are non-sister. And also in the case of on the lower side, this black and this blue is also non-sister, because these are not produced by, suppose this black is not produced by this blue, this black is also not produced by the blue, and doesn't black produces the blue. So that's why these are non-sister. There is no any parent, same parent, that is why these are non-sister. So suppose, look at carefully, this is the chromatids of the two chromosomes. Okay? Now what is happening actually? These are suppose two lower chromatids, and these are the two lower chromatids, and also upper have two chromatids, and this chromosome also have two chromatids. Now these two are sister chromatids, and these two are sister chromatids. So what happened? The crossing over means the genetic exchange of genetic material takes place in between the non-sister chromatids, mean between this chromatid and this chromatid. Suppose right chromosome and left chromosome, right chromosome and left chromosome. So the exchange of genetic material takes place between the chromatid of right chromosome and chromatid of left chromosome, like this. So it will look like a X shape. It will look like a X shape during the crossing over. And the point where the crossing over takes place, that point is called as chiasmata. So here I am showing the chiasmata. Now what is the chiasmata? Chiasmata is the point where the crossing over takes place. That point is called as simply the chiasmata. So this is the crossing over. So crossing over always takes place in between the non-sister chromatids of homologous chromosomes. These are homologous chromosomes and these are non-sister chromatids. So simply the crossing over means exchange of genetic material between the non-sister chromatids of homologous chromosome. So it will look like a X shape. So chromosomes become the thick and short into this Picatinny stage, and the important is the crossing over. So crossing over means the exchange of genetic material between non-sister chromatids of homologous chromosome. This process is called as recombination. This process is called exchange of genetic material. That process is called as a recombination, and pairing of homologous chromosome, that process is called as synapsis. So don't confuse with the synapsis and the recombination. Recombination means the exchange of genetic material between the non-sister chromatids of homologous chromosome. That is called as recombination. Okay? And the crossing over means crossing over means it is a process, and recombination is the term for that exchange of material, because there is a recombination of the two genetic material. That is why we are saying the recombination. Now the next step and fourth stage of the prophase first is the diploteny. Le ZP DD. So now D for diploteny or diplonema. Diplo means two, nema means the thread. So here now the separation of the two threads, that is why it is called as a two threads. So what happens? Homologous means homologous chromosomes start to separate by dissolution of synaptonemal complex, because this synapsis or pairing is done by synaptonemal complex. So their separation will be also by dissolution by the synaptonemal complex simply. So now they start to separate these homologous chromosome, that is non-sister or homologous chromosomes start to separate like this. Suppose this is the pairing, that is the chiasmata where the crossing over takes place. Now they start to move like this and finally they get separated. But in the diploteny stage these are not completely separated, they will get separated into the last stage, that is the dikinesis. So homologous start to separate by dissolution of synaptonemal complex. So that's why in this diagram I am showing very closer these two chromosomes, but in this diagram now they are separated, they start to separate. So they separate except at the chiasmata. In the diploteny stage these are not completely separated, these are separated but except at a point, that is the chiasmata, and that's why they look like this, that is the X-shaped structure. We will observe the X-shaped structure of the chromosome during the diploteny stage. So this is the chiasmata. Now the centrioles is completely migrated to the opposite pole. This is the nucleolus, and now these are moving apart. These are moving apart from each other. Okay? This is moving in this direction, this is moving in this direction. So here they start to separate, but these are not completely separated. These are paired at a point, that is the chiasmata. So that's why the shape is look like this. So now what happens in the dikinesis? In dikinesis these are intact. Now finally in dikinesis they get completely separated. But what happens actually after the separation? There is exchange of the genetic material. There is exchange of genetic material. So in the dikinesis we will see the exchange of genetic material, but here we completed the zygoteny, Picatinny, diploteny, and the first stage, that is the liptoteny stage. Next step in the meiosis first is the dikinesis. We have seen all the 4 stages of the prophase first, that is first one is the liptoteny, second one is zygotiny, then pachyteny, fourth one is diploteny stage, and now the last one stage of the prophase first of the meiosis first is the dikinesis. So what happens actually into the dikinesis? In dikinesis, the chiasmata, now you know that the chiasmata, what is the chiasmata? Chiasmata is the point where the crossing over takes place, that is the chiasmata. So chiasmata gets shifted to ends of the chromatids. Now the chiasmata gets shifted to the end. These are not completely separated. The chiasmata is shifted to the ends of the chromatids, and that process is called as a terminalization. That process is called as terminalization. So terminalization is occurs into the dikinesis. You have to remember this, terminalization occurs in the dikinesis. Simple terminalization in dikinesis. Now another one point is that the nucleolus, because in the prophase or in first stage, that is liptoteny, there is no dissolution of the nucleolus and nuclear membrane. So all the process of dissolution will occurs into the dikinesis step. So nucleolus and nuclear membrane completely disappears, okay, or start to disappear. That is why I am showing in the dotted line. These are disappearing, and spindle fiber becomes the apparent, and these spindle fibers which are not present into the diploteny stage or zygoteny or pachyteny stage, this appears into the dikinesis stage. So that's why I am showing these are the spindle fibers which are appearing. These are not completely formed, but these are appearing in the dikinesis. So important point is terminalization and the nucleolus, nuclear membrane, and the spindle fibers. Now here we completed the prophase first. So basically prophase first have 5 substages, that is Le, ZP, DD, means liptoteny, zygoteny, pachyteny, diploteny, and dikinesis. So here we completed the dikinesis. Up to the dikinesis it is a prophase first. Now the next phase is the metaphase first. Now prophase first is completed, now metaphase first. So what happened in metaphase first? Spindle formation is completed into the metaphase first. So that's why the spindle fibers are complete and they get attached with the centromeres, and chromosomes are arranged. Now we have seen the metaphase into the mitosis. So what happened into the metaphase of mitosis? The chromosomes get aligned at the equatorial plane. So this is the equatorial plane of the cell. So same in metaphase will happen, that is the chromosomes are arranged at the equatorial plane. So these are arranged at the equatorial plane of the cell. This is the characteristic of metaphase. So this is the homologous pair. We have seen the homologous pair. These are the spindle fiber and these are the centrioles from which this spindle fiber arises. Now actually in the diagram I am showing these are separated, but these are not completely separated in the metaphase first. They get separate into the anaphase first. So you have to draw these are closer to the each other. In your note you have to draw closer to each other, these arms. But another one thing is that these chromatids actually exchange their material, because crossing over takes place, and during the crossing over they exchange the genetic material. So these are the non-sister chromatids, these are non-sister chromatids. So crossing over takes place in between the non-sister chromatids of homologous chromosomes. So this is a blue chromosome, this is a black chromosome for our understanding. So what happened? This blue chromosome, this much portion is exchanged with the black, so that's why here is a black, and it gives this portion to the black, that is why here is a blue. Same on the lower side. So these are the important events, that is spindle formation is completed and chromosomes are arranged at the equatorial plane in the metaphase first. So this is the end of metaphase first, simple metaphase. Now important, that is the anaphase first. One point you have to remember that the centromere does not divide into these anaphase first. We have seen in mitosis, in anaphase there is a dividation of the centromere. Centromere splits, that is a characteristic feature of the anaphase in the mitosis. But in anaphase first of meiosis first, there is no division of centromere. Centromere does not divide, and due to this is a reduction division. You know that meiosis is a reduction division, means the chromosome number will become half. So this point is important. Then now what happen in the anaphase? So we will see there are total 4 points. First one is homologous chromosomes start moving toward the opposite poles. Now what happen actually, these homologous chromosomes start to move the opposite poles, means this and this. Next, separation. Now this movement, you know that this movement is due to the spindle formations, due to the spindles, due to the chromosomal spindle. As the chromosomal spindle fiber contracts, they pulls these chromosomes towards the respective poles, and interchromosomal fiber stretches, and due to the stretching they pushes the chromosome towards the opposite poles. So these are the forces, same forces which are acting in the mitosis, that is the chromosomal fiber and interchromosomal fiber. Now second point, separation at the chiasmata is completed into the anaphase first, and that process is called as a disjunction. So this is another important point from the anaphase that you have to remember, that is separation at chiasmata occurs into the anaphase first. That process is called as a disjunction. Now, so these are completely separated, and now these are moving towards the opposite pole. This is moving towards this pole, and this is moving towards this pole. This is the direction of moving of the chromosomes. Next point, each chromosome is with the 2 chromatids. So this is one complete chromatid and this is one complete chromatid. So 2. Don't confuse, don't count like this 1, 2, 3, 4. This is the wrong. I have told in the last topic, that is, this is a complete one chromatid. Suppose like this is a chromosome, structure of chromosome. So this is a complete chromatid and this one is a complete chromatid. So 1 and 2. So that is why each chromosome is with a 2 chromatids, and these chromatids have the exchanged segments. These chromatids have exchanged segments. During the crossing over they exchange the material. That is why this chromatid is with the exchanged material. And one centromere, 2 chromatids and one centromere, because there is no splitting of the centromere. Centromere does not divide in the anaphase first. So one centromere and 2 chromatids at each centromere. This is the third point. Now fourth one, at the end of the anaphase first, half the number of chromosomes gather at the respective poles. When the anaphase 1 is completed, what happens? This chromosome will reach to this pole and this chromosome will reach to this pole. So that is finally, and half the number of chromosomes, because we are considered 2n is equal to 2c condition, means diploid condition is equal to 2 chromosomes. But here in anaphase 1 you are looking that this chromosome will reach at this place and this black one reach at this pole. So here is a 1 chromosome and here is also 1 chromosome. So at the 2 different poles there is a half number of chromosomes, because parent cell have 2 chromosomes, but now at the 2 opposite pole here is a 1 and 1. So half number of chromosomes will get gathers at the respective poles. So it means that in human beings 23 chromosomes will present at the suppose upper pole and 23 chromosomes will present at the lower pole with the exchange segment. So here anaphase is completed. Anaphase first is completed. Now last phase of the meiosis first, last phase of the meiosis first, that is the karyokinesis, because it is a division of the nucleus. Meiosis first and meiosis second. Meiosis first also have karyokinesis and cytokinesis. Meiosis second also have karyokinesis and cytokinesis. So we are completed the meiosis first. In meiosis first there is a karyokinesis. In karyokinesis there is a prophase first, and in prophase first there are 5 substages. So we completed the 5 substages, and now the last phase of the karyokinesis of meiosis first is the telophase first. So now what happened in the telophase? Finally the chromosomes are reached to their respective poles, and as they reach to the respective pole with a exchange segment, there is a reappearance of the nuclear membrane, reappear of nucleus at the respective poles. So chromosomes, and another one point is chromosomes start to again uncoil. These are condensed and become short. Now they start to uncoil to form the chromatin, means which forms the again thread, chromatin network. Then nuclear membrane is reappeared, and nucleolus is also reappear in the telophase first. This is the same which is happening in the telophase of the mitosis, because in the telophase of mitosis there is a reappearance of nucleus, membrane, nucleolus. Okay. And another one important point is that the spindle fibers gets lost, or disappearance of the spindle fiber into the telophase first. So now what happened in the next step, that is cytokinesis. Karyokinesis is completed. Now next step will be cytokinesis first.